Hi folks, I just want to give you a few tips about how to do this virtual lab. It's just like the last video from the pre-lab homework where the guy goes through all the steps on how he does his gel, except this is just a simulation rather than the real thing. So we do everything he does except we do it online. Now I'm going to use level 1 here. I have not actually tried level 2 or level 3. I looked through level 3 a little bit. It seemed very much the same. If you want to do level 2 after you finish your level 1 and you submit the work, if you want to look at level 2 and see what the differences are, or look at level 3 and see what the differences are, I would appreciate that a lot. Thank you. If not, that's totally fine. Just go with level 1, like me. So you can skim this. Most of this stuff you already know from the pre-lab quiz. This is actually really nice information. I suggest you read this because this is going to help you with the lab report assignment later on. These are all the things that we're going to use. Please look at these just quickly so that you do not get surprised when we do the lab online in a moment. Now there are a couple of questions they want to ask you just to make sure you know what this experiment is for and what it's about. Now in this particular experiment, I'm going to tell you right now that the scientist, you are looking for a certain protein which is called CCR5. You are looking for a certain protein which is called CCR5, and you just changed some cells, and you want to see how those changed cells are different from the unchanged cell. All right, so the positive control would be the unchanged cell. CCR5 is produced by the unchanged cell. So you want to, wild type means unchanged. By changed, I'm talking about a genetic change. They use this technique called CRISPR, very sexy new technique, recently discovered, I think it was in the 80s. Oh, that might not be recently for you. You may not have been born yet. But yeah, somebody won the Nobel Prize for CRISPR very recently. But I'll let you figure out which one of these would be the closest to a cell line that makes wild type CCR5. That wild type means unchanged, not genetically altered. So that's the key word there. Negative control. Now, you also want to run a sample down your gel that you know does not contain CCR5. And the reason is, if you see CCR5 in this lane, that means your whole experiment failed because it got contaminated and you need to do it again. That's why we use a negative control, to show everybody that we did not mess this experiment up and everything is working the way it should. And so what here has no CCR5? You can read this and look at the pictures to figure that out. Alrighty. Now looks like okay, so it looks like there are two different kinds of genetic edits that you made. You made an edit called edit 4, and you made an edit called edit 7. And those are the two things that you're going to be looking for in the gel once we finish the gel, to see how much CCR5 got produced, or if that CCR5 got changed in some way. Did it get alkylated? Did it get phosphorylated? You know, that kind of thing. And then you're going to predict. You're going to make some predictions. Okay? Now, I want you to take a look at these things. This long blue line at the bottom represents the bromophenol blue, or the loading dye. And loading dye is very light. Its molecular weight something like 700 daltons, whereas the proteins are all in the kilodaltons, like hundreds of kilodaltons. Or sometimes like 60 kilodaltons, something like that. But anyways, this is a very light molecule. It does not bind to protein. The purpose of this is to go to the bottom of your gel so that you know when you are done. This guy is not done. This guy is not done. This guy is done. And this guy is overdone. If you stop the gel here, look at these bands. They're too close together. You would have a hard time proving any hypothesis, or even disproving a hypothesis. This is even worse. This is good. The bands are as far apart as you can get them, and you may be able to prove your hypothesis or disprove. What about this? You cannot prove, nor can you disprove anything with this, because we don't know where the gel ended, where the electrophoresis ended. The loading die is far off, so just because you don't see a band here, that doesn't mean this protein isn't here. If you don't see a band here, that protein may have gone off, and you don't know how many proteins went off. Even though you know that what was in the ladder, someone could say, hey, maybe you had a protease contaminating your whole thing, and some of these bands represent the proteins of the ladder, of the reference ladder that got cut in half. You know what I'm saying? So you cannot do anything if you let your gel electrophoresis run so long that the loading die went off the gel. That's worthless, totally worthless. Now, I know what you're thinking. You're thinking, how come I only see the reference ladder? Okay, the reference ladder, of course, is the sample that you buy and you load into your gel, which contains known proteins of known molecular weights. And you put the reference ladder in here so that you could have a standard against which you can compare the positions of your other bands. Now you're thinking, where are the other bands? Well, the other bands are invisible because you do not stain them. You can stain them after you're done with this electrophoresis if you want. Then you could see them turn as blue lines, just like this. Or you can not do that. You can stain them in some other way. The reason why we do not stain the other proteins is because once, as many of you correctly observed in a previous lab report, once you add Koumassi blue, it's very hard to go back. It's very hard to take the Koumassi blue out. So if you wanted to use those proteins for some other experiment, like mass spectrometry or something, you would have to remove the Koumassi blue. But that's very hard to do. It's almost impossible. It's not impossible, but it's very hard to do. And it's just not worth doing, usually. So that's why you do not stain them right off. If you know that you're not going to use these proteins for anything further, then yeah, go ahead, stain them with Koumassi blue. But usually you don't want to do that unless you're sure that all you want to do is see the proteins in here, and that's all you want to do. Okay, so you'll answer those questions, and you will check out the protocol. I do not want you to read the protocol extremely carefully, but I want you to look at the protocol to see what we're doing and in what order we're doing it. Okay? I think this is the this is not the protocol. This is the protocol over here. All right. Now, if you are feeling not confident about SDS page even after the prelab, by all means, please read the protocol very carefully. Go back and watch some of those videos again from the prelab quiz. Okay. So, all these things are very clearly explained in the protocol, but I just want to show you a couple of tricks. This drawing is the tube rack, which is right here. It is the top view of this. So it's 1, 2, 3, 4, 5 microcentrifuge tubes, and that's 1, 2, 3, 4, 5 microcentrifuge tubes. So this is not just some decoration. This is actually the tube rack. So you can actually put stuff in here. Like you can move tubes when it's the right time to do so from the tube rack into the heater. All right. Now the heater, you can change the temperature of the heater when it's time to do that, and you can change the time. If you hold the arrow button down with your mouse button, you can make the time move faster. Right? Start and stop. Oh, okay. I don't know how to stop it. So how do you know when this is done? You know it's done when the time is back to 0. So you set the time to whatever you thought it should be set to, as it says in here, and then you'll press start, and then very rapidly the time will go down to 0. Let's say you put 5 hours here. It will take like 30 seconds for 5 hours to go by. So they speed up time here very conveniently. All right. Okay. Now, another thing. These are pipette tip boxes. All right. Most of the time, we'll be using the P200 and the P20. Now, you have to open the box, and then you have to drag the pipette over there, and then you have to drop it. But you see, nothing has happened here. So you have to drop it in just the right place. You see that? You see that? It has taken me, I got lucky this time. I got the tip to go on the pipette my second try. But usually it takes me 5 tries to get the pipette tip to go on the pipette. You just have to figure out where to put it. And it's very much trial and error, right? Now, if you want to change what volume this P200 is set to, you just go click on the volume reading, the volume bubble here, and then you can change the pipette volume reading, right? So this is a P200, so this is 48 microliters right here. The reading is very similar to the real reading that we see in the lab, that we saw in the lab. And then the really funny thing here is that if you want to pick something up, you have to click on it to open it first. So see the blue cap? Let me show you this a little bit bigger. There was a blue cap here that I just clicked and took off. So I'm going to take off that blue cap, and then you got to put this in the liquid there, and then you got to click and hold until it's the first stop. Right? And you could see here, you could see that there's some orange stuff in there. That means you have filled the pipette. Let me show you what not to do. If you hold this a long time, you'll go to the second stop. And see, that's not what you do. You don't want to go to the second stop. This is just like a real pipette. You want to go to the first stop to suck. To spit, then you go to the second stop. All right? You got to open the trash can, and then you got to click on the eject button. See how nifty this is? I mean, this is so cool. Geniuses, right? All right, so now let me do that again. So let me get another tip here. First try, second try, third try. Wow, third try. Nice. And then go to the first stop and let go of the mouse button. And then I got that liquid there. And then if I want to put it in something, I got to open the tube. There it is. I opened it. You see that? And then I go to the second stop. Second stop. I just keep the mouse button down. Do not double click. You just press the mouse button and hold it there until second stop shows up. All right? And you could see that there's liquid in here. That's what I'm going to look for in your screenshot. I'm going to look to make sure that the liquids in here are the correct color. And I'm going to look to make sure that your gel, so this is your gel, you put the gel in the box, you know. I'm going to make sure that your gel has the comb removed and it has certain wells. So you could see the screenshot that I want you to have in the Canvas assignment. I showed you my screenshot, all right? Okay, so I also want to show you this vial. This vial, when you open it, it has a blue top. When you close it, it has a white top. So it's a little confusing in that way, all right? And you always have to make sure you throw away your tip, right? It's going to give you an error message if you don't do that. Now, they're going to ask you to fill the gel with running buffer, okay? Now, because we're not going to do this in real life because of the pandemic, I got to just tell you, you got to fill this with the running buffer first, right? You got to fill the gel cassette with the running buffer. I want to tell you why you do that, all right? There are 2 reasons why you do that. Right? The first reason is when you fill the gel cassette with the running buffer, you soak the gel with buffer. And that's good for the gel because if there's any dry spot in here or some air bubble, you want to know. And you want to also prevent any dry spots from occurring. And so this thing has to be soaking in buffer. If you find a dry spot or some bubble that somehow you didn't notice before, you have to throw away the gel. You cannot be running a gel that has bubbles in it because then the protein, as it goes down, if there's a big bubble there, it's going to get stuck and it's not going to go to the place where its molecular weight says it should go. Right? Also, you want to fill this with running buffer because The whole point of gel electrophoresis is to use the electric field, positive charge down here and negative charge up here, to force proteins coated with SDS down there. And you cannot run a strong enough electric field through air or even through the gel itself. You need liquid that, like water, that has ions in it, like buffer, to set up that electric field so that it's strong enough to and evenly dispersed enough to make your proteins move. Let's say you did not use running buffer. Could you still set up an electric field? Well, I mean, if you put a wire just directly on the gel, which we do not do, and another wire directly on the gel, perhaps you could set up such an electric field, but you probably cook the gel right where the wire touches the gel, or something like that. Okay, so that's another reason why we add the running buffer to the gel cassette is we want to sink the samples without any air bubbles into these wells. So if we did not fill the cassette with a running buffer, then when you add your samples, yeah, they would sink into the wells because there's just air there, but there may be bubbles, and doing that in the presence of buffer, where all the wells are full of buffer, just watery, salty water and some pH buffer there, that helps the air to leave as bubbles. And if it doesn't leave, you can just tap it out if you need to. I'm trying to remember, how important is it that those bubbles get tapped out? I kind of forgot. That's a problem with doing this online rather than in real life. Okay, now, so after you load the gel, you're going to have to put the running buffer again in here. You're going to put the running buffer in the tank box. And the reason for that is that so these electrodes, you can see the electrode, you cannot see that. So these electrodes are not in the cassette. Actually, I take that back. So you have to have the running buffer in the cassette for a reason which I forgot. But you have to have the running buffer in the gel box for a reason that I forgot. But you may think by looking at this that the negative side of the electric field is on the left and the positive side of the electric field is on the right. Well, that is not the case. The cassette has a wire that runs along the top, and that wire connects to the negative electrode. So the electric field is set up here, not here. The picture is a little deceiving. And the positive electrode connects to a bare wire. Usually it's made of platinum. That's at the bottom of the cassette. And so your electric field is truly set up with negative on top and positive on the bottom, not negative on the left and positive on the right. All right, so have no fear. We're not smoking crack here. This is real science. All right, so that's the trick with the running buffer. I forgot why. Okay, but that's when you do this in real life in some research lab, you will understand immediately why we need running buffer on the outer gel box, because it'll be right in front of you. I just forgot exactly why that is. So you're going to end up also needing to connect the electrodes. All right, so the software will tell you when the electrode is connected. Usually there's a message that shows up in the middle. It says, oh, your electrode has been connected. So red goes to red and black goes to black. All right. I don't know why it's not telling me now, but it's important for you to have the. You see this? You see this? Oh, here. The power supply has a cable connected to the cathode. All right. So, but you see this. You see how? Let me make this a little bit bigger. And so here's what I found when I did this. You see there's the you could see the exposed brass plug here. That's no good. Your brass plug has to disappear, and it disappears when you've got this properly inside the power supply. If you try to run the gel with the brass plug not buried inside the power supply, then nothing's going to happen, and you're going to get some kind of error message. All right. This guy also has to be plugged in right there. So it's good that the brass plug has disappeared. That means it is buried inside the barrel where it's supposed to go. Okay, so I think those are all the tricks that I encountered in here. Oh, no. There's something else. So you're going to end up having to centrifuge something. All right. Okay, so yes, that's true. You do have to open the centrifuge, and then you can put stuff in there. And then when you centrifuge, so you got to close this tube before you put it in the centrifuge. When you put something in the centrifuge, it's not okay to just close centrifuge and assume that everything's going to be fine. That's not okay. You have to click on the centrifuge, and you have to arrange these things so that they are opposed to each other in a symmetric fashion. So you see, if I have two things, they have to be directly opposite each other. If I have three things, they have to be in some kind of radially symmetric fashion. So everybody is 3 spaces apart. If you don't do that, in real life, the centrifuge will be damaged as it runs because it will rock back and forth like a clothes dryer that's unbalanced. So you're going to have to figure out some way to arrange your samples so that they are evenly spread out in some kind of symmetrical way. This is called radial symmetry, right? I think this is called radial symmetry, but I am not sure. And then you will click on check balancing, and then your software will verify that for you. You got to close the centrifuge before you run the centrifuge. And centrifuge is very similar to the heat block in that there's a timer, and the timer runs down really fast, faster than real time. I think that's all that I needed to show you in here. Please do the lab work and have a good little time doing it. Warning. Just like real lab work, the virtual lab work is picky and it's boring. I had to take a nap while I was doing this, in the middle, because I was falling asleep. And you know what? In my first lab job, I also had that experience. I was drying DNA using this little air blower in these little micro centrifuge tubes, and I was falling asleep while doing it. And then the boss was like, "Dude, you're falling asleep while you're doing that." And I was like, "Oh, my bad." So that's it. Yes. One more thing. You're going to need to give me a screenshot. Your screenshot should be just like this. It's got to show me that you've got all these things, and it's got to show me that. I guess that's all I could see. All right, so it's got to show me that you've got all these things. You got to type me a little message here. "This is Glenn," or "This is," who are you? Are you Jan? "This is Jan," or whoever you are, right? And then I want to see another tab here in the back that shows your email address, so I know it's you, and I know that you didn't just get this from your lab partner by email and you just threw it up here in the canvas thinking that would be okay. Right? I really want you to do this lab work. I want you to experience all this picky little stuff and also the drudgery, because that's part of science. You know, it's what we have to do to wring the secrets out of nature. She doesn't give them up easily. You got to, as a scientist, you got to coax them out with very meticulous care. Thank you. Bye.